Scientific research is a strange endeavor. You spend years working on a project. You carry out experiment after experiment. You collect data, you double-check all your calculations, and then package it all up with an introduction and a discussion into a product called a research paper, and then you just throw that out into the world, actively encouraging anyone around the whole world to read it, to make what they will of it, and to actively try to prove you wrong. Sometimes those people go on to become your collaborators, and sometimes you never ever meet them. Gregor Mendel published his Versuche über Pflanzenhybriden in 1866, his most famous publication, and he died in 1884. It wasn't until almost 2 decades after his death that other scientists around Europe and the world began to read his papers and pick up where he left off to create the field of genetics. That's not really so unusual. Research is a process that does not always run smoothly. Sometimes it can take years for an idea to bounce around between the right group of people until at some point, quite to the surprise of everyone involved, it turns into something new, something shocking that changes the world. And so it was, in 1987. A PhD student called Yoshizumi Ishimo, working at the University of Osaka, published a paper and sent it off into the world. The paper was titled, Nucleotide Sequence of the IAP Gene Responsible for Alkaline Phosphatase Isoenzyme Conversion in Escherichia coli and Identification of the Gene Product. Not exactly a title that would make you think a revolution lies in this paper. Alkaline phosphatases are important in maintaining the phosphate balance of the cell, and they were the main focus of the research, which was essentially to sequence a gene encoding an enzyme involved in alkaline phosphatase production in the bacterium Escherichia coli, more commonly known as E. coli. But in the process of sequencing that gene, and I will emphasize in the 1980s, sequencing a gene was a serious undertaking worthy of a PhD. During the process of sequencing that gene, Dr. Ishimo found something else. Something unexpected. Just after the gene, physically in that genome, he found something that was not what he was interested in, but he did describe it in the final part of the paper. He wrote, an unusual structure was found in the 3' end flanking region of the gene of interest. 5 highly homologous sequences of 29 nucleotides were arranged as direct repeats with 32 nucleotides as spacing. What he had shared with the world was the first description of an intriguing phenomenon that will get picked up and explored by scientists around the globe. These repeating sequences seem to be found in most bacteria, and between the repeating units were little pieces of DNA that matched known viruses. Digging deeper, researchers found that these repeating DNA elements are managed and created by a set of bacterial proteins. The repeating DNA units came to be known as clustered, regularly interspaced, short palindromic units. The proteins that managed and created these clusters were cluster-associated proteins, or Cas proteins. There turned out to be many of these Cas proteins. They were given numbers as they were discovered, Cas1, Cas2, Cas3, and so on. By 2020, we were decades into the existence of this fairly niche field of research, interested by then in what had become to understood as a bacterial immune system, protecting the cell against viruses by integrating small chunks of viral DNA into basically a memory bank for the cell to know what to look out for and destroy in future. The next year, in 2011, a small side session was held to discuss these bacterial immune systems as part of a bigger microbiology conference in San Juan, Puerto Rico. And at the sidelines of that session, as fringe as could be the side of a side session, something happened that would change the world. A French professor in Germany called Emmanuel Charpentier approached an American professor who had grown up in Hilo, Hawaii, but worked at UC Berkeley, Jennifer Doudna. Charpentier felt that their 2 research teams between them had just the right mix of expertise to tackle a project on her mind, solving the function of a particular protein that was associated with these clustered, regularly interspersed short palindromic repeats, or CRISPR, as they're known. This protein, Cas9, was little known at that point, but in just 9 years, Doudna and Charpentier would be accepting a prize awarded based on one of the easiest decisions of the Nobel Committee for their work to solve the function of CRISPR-Cas9 and ushering in a new age of unlimited possibility to engineer genomes as we please. Welcome to Modified. I'm your host, Dr. Orlando Delange, explant scientist and current science teacher. This podcast is my attempt to guide you through the science of GMOs and hopefully just enough of the history and the social context to give you a path forward in your own thinking. And if all else fails, I hope you'll enjoy some of the surprising stories and fascinating facts in the world of genetically modified plants. This is episode 16. CRISPR changes the world and unlocks GMO 2.0. If this is the first episode you're listening to, because maybe you saw the word CRISPR and thought, hey, I've heard that, I want to check that out, well, welcome. Happy to have you here. You may find that I dive pretty deep into genetics and into the world of GMOs in this episode, and so you may want to go back to episode 1 and enjoy the journey to get here, because this is the last episode of season 1 of Modified. In this episode, I'll explain what CRISPR-Cas9 actually is and how it works, and I'll explain what I mean by GMO 2.0 and why I don't think it's silly to predict a wave of CRISPR-derived GMOs in the next 5 years. But maybe a good way to tee off the main body of this episode is to hear the words of Emmanuelle Charpentier herself, interviewed in 2022. I will spare you me trying to do a French accent. She was asked, what is the greatest benefit to humankind of your research? And she had this to say, after first explaining possible applications in medicine. There are also great implications in plant biology, in the production of plant crops, specifically in the world of global warming and global climate change, the necessity to be able to engineer crops that can sustain the changes that are meant to be encountered in the very near future and even in the long-term future. In the life of today, we benefit so much of great well-being thanks to a number of technologies like improvement of food and medicine. CRISPR has a great impact on this. That's why it has impacts on humankind. So CRISPR from the get-go was recognized for its possible applications in crop biotechnology, and I can back that up with some personal experience. So let's talk a little bit about me. In 2010, I started my PhD at the Ludwig Maximilians University of Munich, and the goal handed to me for my first project was test the idea of creating a programmable DNA endonuclease using the DNA binding domains of the bacterial proteins that our group specialized on. Those proteins are called TALE, T-A-L-E. It's an acronym. And so we decided to add an N to the end of that acronym to represent the word nuclease, and then you have T-A-L-E-N, talons. Google talons, and you'll still find plenty of material about them. Talon technology got so much interest because the idea of a programmable endonuclease was an already incredibly hot idea in the world of biotechnology, and so a lot of people were desperate to see if these new talons could finally deliver what had been tried since the 1990s with only limited success using different proteins called zinc fingers. But let me back up. What is a programmable endonuclease? I just threw that word out there. An endonuclease is a protein that can cut DNA in the middle of a DNA molecule. Nuclease is a more general word for any protein that can cut DNA. You may remember that DNA is an acronym for deoxyribonucleic acid, so nuclease references that nucleic acid part. The hope is that a programmable endonuclease could allow you to make precision cuts in the DNA of any genome, and that is the first step in then permanently changing the sequence of DNA bases at that genome position, a process known as gene editing or genome editing. There are many nucleases in the world. Some just chop up all DNA into pieces randomly. That can still be useful for some sorts of goals, both natural and technological. And there are other nucleases that are called restriction endonucleases, or just restriction enzymes. They were the ones that drove the birth of recombinant DNA technology and the race to create GMOs back in the 1970s. They are still very commonly used, but they are not programmable. Each restriction enzyme has a single built-in target DNA sequence that it binds and then cuts at that position. Generally, those target sequences are about 6 nucleotides long. Any given 6 nucleotide sequence would, by pure chance, appear thousands of times in any large complex genome, like a plant or animal genome. So restriction enzymes are not a useful tool if your goal is to try and engineer genomes. So as the 70s became the 80s and then the 90s, more and better information became available about the DNA sequences of all sorts of interesting genes. And a lot of that interest was focused on human genes that may play a health role. Perhaps mutated versions cause known diseases, but if you could repair that mutation, you could cure the disease. And there was lots of interest in plant genes that could be tweaked to create improved crop varieties. So there was a real desire to have some way to go in and do genome engineering, but this knowledge was somewhat useful because no tools were available that could actually facilitate this. So for those decades, the 80s and 90s, a lot of money was poured into making use of the tools we did have, namely to piece together engineered DNA molecules in a lab and then randomly insert that DNA somewhere into a target genome. That is the process we call transgenics. That is the process used to make all the GMO crops covered so far in this series, and all of the GMO crops that are currently commercially available, at least outside Japan, to foreshadow the very end of this episode. But that whole time, so we're talking here about the 80s, basically through to the 2010s, while funding was pouring into academic research to study and use transgenics, and while investors were happily funding companies to develop products using transgenics, the dream of a programmable DNA endonuclease, and therefore of genome engineering, did not die. And that's why, when a new type of DNA binding proteins called tails were first described in a scientific paper in 2009, a lot of research groups took notice. I will spare you a lengthy treatise in this podcast, but out of pure vanity, I will link in the show notes a review article I wrote about tails, talons, and their uses in plant biotechnology, and how, at the time, we tried to spell out the possible advantages that talons still had versus the nascent CRISPR technology, since we wrote that article in 2013. Needless to say, CRISPR won that argument. The cliff notes are that tails are large proteins, and most of their bulk is a set of repeating units, and each of those repeating units binds to one DNA base, and there is a single amino acid that differs between each of the repeating units, and that gives a specific DNA binding preference to that repeat, using a specific code that links that amino acid, that variable amino acid, to a specific DNA base. So if that variable amino acid is an isoleucine, the repeat will bind to an A in the DNA. If it's a glycine, it will bind to a T. If it's an aspartate, then it will bind to a C, like that. So say you wanted to target a 15-nucleotide DNA sequence that appears in a gene you're interested in, you just have to go to your lab freezer where you have stocks of different DNA pieces that encode tail repeats, each of which have been pre-modified to have molecular cloning sites attached to their ends that will allow you to assemble them all into a full-length tail gene in a one-pot reaction, given the right mix of temperatures and enzymes to assemble everything together into a large DNA vector called a plasmid. And then you spend about a week sifting through the different reaction products to find one that was correct. And so basically, as long as you've done a lot of upfront work to get all the pieces prepared for this process, you could create a totally new designer tail, or D-tail as they're known, in about a week. That was a sensation. That was newsworthy, and it was investment worthy, with new companies springing up around this technology and established companies sending teams of lawyers to negotiate a license to use the technology. But so far, what these proteins had evolved to do over millions of years was just to act as transcription factors, not as programmable endonucleases. The TA in tail is short for transcription activator. Naturally, tails are delivered by invading bacteria into plant host cells, where they then navigate to the nucleus and bind to the specific gene that has a DNA sequence matching the pattern of repeats that make up the most of the bulk of the tail. Then, when it has found its target gene, it will recruit RNA polymerase to initiate transcription, the first step in gene expression. In other words, it will activate the gene switching on gene expression. And the most famous example of this, what it does is help the bacteria by switching on production of a specific gene that encodes a sugar transport protein that pumps sugar out of the plant cell into the surrounding extracellular matrix where the bacteria are feeding and replicating, essentially forcing the plant cell to feed the bacteria that are attacking it. So yeah, tails naturally are transcription activators. Switching on genes is useful to some extent. So just being able to rearrange natural tails to make designer tails is useful. But as I mentioned, it was really programmable endonucleases that were seen for decades as the real prize. But now I should explain why. What is actually so useful about being able to cut DNA? Well, it's not actually the cutting of the DNA that's so interesting. In fact, if you just cut DNA inside a cell, really cut all the way through both strands, severing a link in the chain of the long DNA molecule called a chromosome, that is actually fatal for the cell. It cannot be tolerated. And so cells, in all organisms, have a DNA repair kit in the form of a range of enzymes that can work together to carry out a few different types of repair, healing that break. The simplest approach, in terms of the number of DNA parts involved, is called non-homologous end joining, or NHEJ. This literally means sticking the two ends together, and in practice this involves either chewing back the DNA a bit to create very short matching DNA sequences that can be stuck together, or just chewing back one strand till you find an A, and then you chew back the other till you find a T, to give you an example. And then you can stitch those together using homologous base pairing, which if you remember is A to T and G to C. Alternatively, a few DNA bases can be added randomly at the break site, in the same way creating a tiny piece of overlap, so for example a G on one strand and a C on the other that can bond together to form a GC bond, which can ultimately heal the entire double-strand break. We describe non-homologous end joining as error-prone, meaning that it will heal the DNA cut site, but it will almost always change the DNA sequence in the process, introducing short deletions or insertions of random DNA when compared to the original sequence before you made the cut. That is actually very useful from a biotechnology perspective, because if you target a gene with one of those cuts, then the process of healing the cut will introduce mutations into the gene that will in most cases disrupt the function of the gene, essentially breaking the gene. So let's give a real-world example of why that can be useful. Sickle cell disease is a brutal genetic disease. It mostly impacts people of West African descent, and it is caused by a mutation in the gene encoding hemoglobin, the crucial oxygen-carrying protein in our red blood cells. More specifically, it is a mutation in the gene encoding adult hemoglobin. There is a separate gene in our genomes that encodes a version of hemoglobin expressed while we are in the womb. That's fetal hemoglobin. In adults, that fetal hemoglobin is not produced because the gene is switched off by a transcriptional repressor protein. As long as you can get into the blood producing bone marrow cells, you can use a programmable endonuclease to make a cut in the gene that encodes that transcriptional repressor protein. Then you can let non-homologous end joining do its thing to mutate the gene and basically break it. Now, without that transcriptional repressor, the fetal hemoglobin gene switches back on again, even in adult bone marrow cells. And now the bone marrow cells start producing fetal hemoglobin alongside the dysfunctional adult hemoglobin. That's not enough to totally remove sickle cell disease symptoms, but enough to vastly improve them. And that is exactly the mechanism behind the first and only CRISPR-Cas9-based gene therapy currently available in the US, a therapy sold under the brand name Casgevy. And if you think about crop plants, one of the big uses would be to mutate, or as is generally said in plant genetics, knock out a gene that encodes a protein that is targeted as a receptor of pathogens. It is a common feature of pathogens in all organisms that they need something to latch onto so that they can stay attached to the host cell long enough to do their things. This is also true of viruses. For example, the SARS-CoV-2 virus that causes COVID-19 latches onto a human protein called ACE2. The same basic mechanism is crucial for bacterial pathogens, fungal pathogens, and so on. There has to be some host molecule, usually a protein, and usually one that sticks out of the cell membrane, for the pathogen to latch onto. In all cases, these host proteins, what we often refer to as pathogen receptor proteins, did not evolve to offer an attachment site for pathogens. That would make no sense. They have some other natural function for the cell. But from the perspective of a farmer or plant breeder, whatever purpose that pathogen receptor protein actually serves in the normal healthy function of the plant, is probably outweighed by the damage done by the pathogen being able to use or co-opt that protein to invade the plant cells and ultimately destroy a field of the crop plant. So being able to knock out a pathogen receptor protein in plant genomes is a really desirable use of programmable endonucleases. But so far, I've only talked about one of the possible repair pathways for the cut produced by a DNA endonuclease. There is another option that is even more exciting: homology-dependent repair, or HDR. In this approach, a second DNA molecule is used as a repair template. And the real trick is that as long as the repair template has a good amount of perfectly matching DNA sequence at its beginning and end, the middle can be different to what was originally in the targeted DNA molecule where you made the cut. It's kind of a Trojan horse approach. Trick the cell into thinking that the repair DNA you've given it is just a perfect match that it can use to close the DNA break that it needs to repair. But in the process, it is actually introducing a mutation. But in this case, the mutation would not be random. It would be a specific DNA change designed in a lab and built into the repair template DNA that is delivered into the cell alongside the programmable endonuclease. This approach, where you can change the DNA sequence however you want to, is called gene editing, or done at scale, genome editing. Sometimes gene editing is also used as a term to describe the random targeted mutations described previously. But most of the time, if you hear the term gene editing, it means these specific DNA changes introduced at specific sites in a given genome using homology-dependent repair. These DNA changes are technically mutations, because that is a word used for any change to a DNA sequence. But one of the uses of gene editing that is most exciting in terms of human health is to use it to fix inherited genetic mutations that cause disease. So to connect back to sickle cell disease, it would be seemingly much better, more elegant and better for the patient if you could just go in and fix the mutation in the adult hemoglobin gene rather than this convoluted approach of breaking a transcriptional repressor to allow expression of fetal hemoglobin. So you might think, well, why wouldn't you just do that? Why don't you just go use gene editing? Why did they use that convoluted approach? Well, here's the catch. Gene editing is actually a lot harder to do because cells generally prefer the approach of non-homologous end joining over homology-dependent repair, and because at a technical level, it is difficult to get that repair template DNA into the cells where you need it to be in order for gene editing to work. So that pours a little bit of cold water on the excitement on the new era of gene editing that CRISPR has ushered in. It is still something very difficult to do, even if we have a perfect tool for at least the first step in gene editing. But back in 2009, even that first step was basically non-existent, because we had no programmable endonucleases, apart from the fairly clunky zinc fingers, to be able to make the DNA cuts that were the first step in gene editing. So that's why when tails appeared on the scene, multiple research teams scrambled to work out how to turn those tails into talons. That's the nuclease version. And that was done at the DNA level by fusing a non-specific DNA nuclease domain onto the end of the tail gene. Basically, take this thing that doesn't naturally have the ability to cut DNA and add on an extra domain that will allow it to do that. Much to my dismay, that was successfully demonstrated just a few months into my PhD, but not by me. Deflated, I moved on to a different project that was more linked to the natural role of tails in causing devastated crop diseases called bacterial spot and bacterial wilt disease. But just a couple of years later, while I was midway through my new project, I read a research update that made me very happy that I hadn't hitched my academic success to talons. That update was a description in probably Nature or Science magazine describing recently published research into a system called CRISPR-Cas9. And after spending just a minute or two reading and then cross-checking the actual research article authored by the labs of Jennifer Doudna and Emmanuel Charpentier, I knew that this was the future. Not just me, really everyone did. The paper is titled A Programmable Dual RNA-Guided DNA Endonuclease and Adaptive Bacterial Immunity, and I've linked it in the show notes. It is open access, so you don't need to pay to read it, which is good because I think it's an important document for the history of humanity. If you've listened to many episodes of this podcast, you know I generally shy away from hyperbole and all the smoke and mirrors of the tech world, whether that's biotech or regular silica-based tech. But CRISPR really was and is a world-changing technology. I was not the only one to immediately say how huge this would be. CRISPR got a massive and rapturous reception from the entire biotechnology community and garnered headlines around the world. Doudna and Charpentier got the Nobel Prize just 8 years after publication of that paper. That sort of speed is very unusual, and it's a testament to how quickly CRISPR has reshaped the field and for the first time made gene editing feel within reach. CRISPR is the shorthand that's often used for CRISPR-Cas9, the combination of the Cas9 protein and a guide RNA that together work as a programmable DNA endonuclease. The Cas9 protein itself cannot do anything until it is brought into close contact with DNA, and then when that has happened, it will make a cut at that site in the DNA. To get the Cas9 protein to the DNA, it partners up with a guide RNA molecule. That is a short molecule made of DNA's molecular cousin, RNA. The guide RNA physically embeds within the Cas9 protein, but a 20-nucleotide region of the guide RNA, called the spacer, is exposed and is basically able to scan along the genome in the target cell until it meets an exactly complementary 20-nucleotide DNA sequence. At that point, once the guide RNA has found its matching DNA sequence, the Cas9 protein gets stuck in place long enough and securely enough to do its thing and make a DNA cut. So all you have to do to get Cas9 where you want it is to modify those 20 nucleotides of the gene encoding the guide RNA. The Cas9 protein remains the same. And since guide RNA genes are so short, they are cheap and easy to chemically synthesize, meaning anyone can sit down and order one for maybe $100. You'll get your own guide RNA gene in just a few days. You can also use laboratory methods to modify that 20-nucleotide spacer region of the guide RNA if you happen to have a guide RNA gene already lying around the lab, and that is quick and easy, taking just a few days. So that is the huge advantage of CRISPR. It is quick and easy to make a guide RNA gene that you can package with Cas9 to deliver into a cell to target Cas9's activity to any gene you want. And that all takes just a matter of days compared to the weeks required to create a TALEN. It may seem a minor logistical advantage, but it's one of several that all add up. Another advantage is that the targeting system is based on RNA, which uses the same molecular matching system as DNA, homologous base pairing. In practice, if you know what 20-nucleotide DNA sequence in a target genome you want to target, then that 20-nucleotide DNA sequence is actually the exact DNA sequence you need to insert into your guide RNA gene as the spacer, because that DNA will be transcribed to produce a complementary RNA that then can go search for its complementary DNA, which would be the same sequence as the original DNA used to produce the RNA. What this means is that it's incredibly easy to design guide RNAs, a lot easier than designing TALENs, which requires some nuanced understanding of the protein to DNA code that underlies the function of TALENs. Almost no expertise is required to design guide RNAs. Almost immediately after CRISPR-Cas9 was first described, free web tools popped up that make it as simple as defining what gene you're interested in targeting, and it will spit out for you a whole set of guide RNA sequences you can use, and even companies offering to just send you the guide RNA gene that would do what you want it to do, as long as you tell it what gene you want to target. And in the process of designing the guide RNA, these tools will check against a computer database of the whole genome of the target organism to ensure that the guide RNA sequence being designed is totally unique within the genome. Another advantage is that CRISPR-Cas9 just works incredibly well, generally better than TALENs, meaning it cuts more efficiently, and it is about as good at avoiding mistakes where it accidentally cuts a similar but slightly different sequence, what we call an off-target. Those off-targets, they do happen and they are a concern. They tend to happen at very low rates. And after about 15 years of CRISPR being on the scene, modified versions of the Cas9 protein are available that have even better performance in regards to off-targets. And another logistical consideration is that Cas9 is a single protein. What I've yet to mention is that to get tails to serve as nucleases, basically to become TALENs, you have to rearrange them as a pair of proteins facing each other, each with a nuclease domain that cuts one DNA strand each, together creating the complete break in the DNA molecule. Each TALEN protein is big and bulky, and it is a logistical problem to get big, bulky proteins into cells, especially if you want to deliver that as protein, not as DNA encoding the protein. The advantage of delivering proteins directly into the cell is there's then absolutely zero risk that endonuclease somehow gets integrated permanently into the cell. Proteins by themselves are inherently unstable molecules within cells. There's no biological mechanism for a protein to become permanently integrated into a cell. Whereas DNA, depending on the delivery method, may well stably insert itself somewhere randomly into the genome of the target cell, making a permanent DNA change that is just a side effect of the gene edit you are actually trying to make. Keep that in mind because it's one of the keys to the biggest selling points of using CRISPR in crop engineering. Cas9 is a fairly bulky protein itself, but it's just one protein that you have to get into the cell, not two. And there are also modified versions of the Cas9 protein that are smaller and easier to deliver as a protein into cells. So CRISPR-Cas9 was just like a faster, better, stronger version of Talon's that felt in reach to really anyone with some basic knowledge of molecular biology, and scientists immediately saw all those advantages, took it, and ran with it. Most of that metaphorical running originally happened in academic research labs, using CRISPR-Cas9 to modify genes as part of molecular genetic research programs. Academic researchers were free to take CRISPR-Cas9 and run with it because they didn't need a license. As you might expect, CRISPR-Cas9 as a tool for gene editing was immediately patented. However, even though the patents were immediate, they were unfortunately multiple. So what I haven't mentioned so far was that the Doudna-Charpentier team filed a patent once they published their discovery, but so too did a team at the Broad Institute, which is affiliated with MIT and led by biotechnology professor Feng Zhang. You see, the Zhang lab had published a demonstration of the use of CRISPR-Cas9 in eukaryotic cells just a few months after the original work by Doudna and Charpentier. A pretty classic situation of two researchers ending up doing a similar thing and publishing at almost the same time. Doudna and Charpentier were CRISPR-Cas experts, but not so much biotechnologists. They did see the potential of their discovery, and they tried to demonstrate the use of CRISPR-Cas9 as a programmable endonuclease in their original work, and they used that to file a patent. But Zhang and his lab were more experienced in recombinant DNA methods and had seen the possible applications of CRISPR-Cas9 based on some research into natural CRISPR systems published earlier on in 2011. So they were basically working on a very similar idea. They didn't really care about the natural CRISPR system, but they saw this idea of how you could use Cas9 as a programmable endonuclease, and they published that just a few months after the Doudna-Charpentier paper. The result of this was a 10-year legal battle that was only resolved in 2017, actually in favor of Zhang and the Broad Institute, a decision that has been reaffirmed multiple times, including as recently as 2026. And as you might imagine, that legal battle has significantly held back commercial investment in and applications of CRISPR-based technology. Despite that, many companies pressed ahead with R&D, even while they weren't really sure who they would have to pay to get a license to be able to use the technology inherent to their approach. By now, that obstacle is cleared, and it's even easier in that, as I mentioned earlier, multiple variants of Cas9 have now been described from different bacterial species, or created through deliberate modification of the Cas9 gene in labs, producing a range of Cas9 variants and alternatives that have their own patents. So anyone looking to use Cas9 or one of these variants commercially has a range of different options of potential partners to purchase a license from. So there are a range of options, but what actually are the products that people are trying to work on? Well, I mentioned human gene therapies. But this is a podcast about crops. What are crop scientists in universities, government research labs, and companies trying to do with CRISPR? First, the general theoretical answer about what you can do with CRISPR that you can't do with general transgenics. Well, one thing that you can do is go in and alter genes that are in the plant. For example, edit just a few DNA bases to change the part of a gene that encodes the active site of an enzyme that might allow the plant to better tolerate drought or heat stress. So remember that there's now been decades and decades of what we call molecular genetic research into plants and their genes and genomes. So we know of all sorts of genes that could be kind of very powerful if you made a slight DNA change to them. Maybe you know that there's a wild relative that's more drought resistant. And if you could modify the version of the corresponding gene in the crop plant to mimic the version that's in that wild relative, you could probably get some boosted drought resistance. So there's all sorts of ideas like that floating around, just waiting to be implementable with the right tools. That is not the sort of thing you could do by just inserting a new gene. You would just be layering something new on top of what was already exists inside the crop, which sometimes might be useful, but sometimes having both versions simultaneously expressed and functioning will kind of ruin the effect you're trying to achieve. But there is another very significant reason that crop scientists were immediately excited about CRISPR-Cas9, probably the biggest reason. If you can get the Cas9 and the guide RNA, so that's as protein and RNA, not as DNA encoding those things, get them into callus cells. Those are those induced plant stem cells that are then used to regenerate a whole plant. So if you can get that Cas9 protein and the guide RNA into callus cells, and the Cas9 protein and guide RNA can work together to get to a gene of interest, break that gene, or even better, if you can get the Cas9 guide RNA and a template DNA molecule into the cells and let them work together to edit the gene of interest, well, after that gene edit, the cells will continue to divide. After a few hours, the Cas9 protein and the guide RNA will be destroyed because proteins and RNA are for the most part short-lived molecules, and cells are full of enzymes that will destroy RNA and proteins, while new ones are churned out to take their place. Except, of course, the cell doesn't have the genes to make the Cas9 protein and the guide RNA because they didn't come from the plant. They were literally injected or inserted into the cells. And the template DNA, it will also be destroyed because, in general, the template DNA that we use in this sort of experiment is a short linear DNA molecule. So that's kind of naked DNA, not the huge long protein packaged DNA molecules we call chromosomes. And those short linear DNA molecules, similar to RNA and proteins, are inherently unstable in cells. They will be chewed up and destroyed within 1 or 2 cell generations. It's very unlikely, but in rare occasions that template DNA might get inserted into the genome of the plant cell. But that would happen very rarely, and it would be very easy for the crop scientists to check for this and exclude any such rare occurrences from what would be the tens or hundreds of developing calli being created in parallel to be sure of getting at least one good one. So as the callus develops into an adult plant, the Cas9 protein, the guide RNA, and the template DNA are all destroyed. But what does remain is the edit in the target gene in the actual genome of the crop plant. That is a permanent change that is passed down through each generation of cells and the developing plant, and then to all the progeny of that plant. And what to make of those progeny of the gene editing experiment? At the genetic level, they contain no outside DNA. No genes that were not generated from within the plant itself as part of its evolutionary journey, just slightly tweaked versions of those genes. Not really any different to the general genetic variation that can occur in the course of evolution, and the more accelerated process of artificial selection that has been used for millennia to produce crop plants. Would that be considered a genetically modified organism? Well, many companies are banking on the answer to that question being no. What that would mean in practice is a huge reduction in costs, as these crop varieties would be treated no differently to any other crop variety developed using non-GMO methods. That is to say, they would be assumed safe and require no regulatory approval at all before being sold. And they would be allowed to be sold in jurisdictions that currently totally ban the sale of GMOs, such as most countries in the EU. Well, unfortunately, if you're hoping into a deep dive into all the regulation around CRISPR crops, you'll have to wait until Season 2. This episode is already long enough, but I can give you a brief summary. In the last 5 years, countries around the world have taken this question very seriously, and most have basically landed somewhere in the middle, agreeing to relax regulations on crops created using CRISPR-Cas9 gene editing, especially if it's done in a way that leaves no trace of the Cas9 or guide RNA genes in the plant, as I was just describing. In the USA, the most relevant regulatory agency, the USDA, actually created a totally new regulatory framework that would have wiped away most regulation from crop varieties developed using CRISPR. But a judge in 2025 vacated that regulatory framework based on process issues. So that has added further confusion and delay to the original confusion and delay caused by the patent battles. And so it's maybe not surprising that despite the immediate rush of excitement to make use of CRISPR as a tool, going back more than a decade now, what I've been calling GMO 2.0, which is CRISPR gene editing or crops varieties created using CRISPR gene editing, have not really hit the market. Despite that, so as far as I can work out, there are 0 CRISPR gene edited crop varieties for sale in the USA. There are several in the works, though, and there is a CRISPR gene-edited crop for sale in one country, Japan. The Sicilian Rouge high GABA tomato, created and sold by Sanatech Life Science. Much like the purple tomatoes covered last episode, this is a variety modified to produce higher concentrations of chemicals with purported health benefits. So maybe to wrap up this episode, in episode 11, we met the first ever GMO crop, a tomato, and we can end here with, I believe, what is the first commercially available GMO 2.0 CRISPR gene-edited crop, another tomato. That is it for Season 1. I've had a blast putting it together and I've learned a lot, not just about the history of science and GMOs, filling in a lot of gaps in my knowledge as I research each episode, but also about how to do audio editing and video editing, as I've also started to do more video content, and how to generally create a podcast. I feel very far away from the standards that I would hope one day to achieve, and I have a vague notion that at some point in future I'll come back and redo this season, but really stepping everything up a notch. In the meantime, we will keep moving forward into new territory. Season 2 will be putting the basic science of GMOs a little bit more in the background and bringing the economic and social impacts of GMOs more into the spotlight. But there will still be a mixture of both, so don't worry science nerds, I won't let you down. But the thing is, I will have a lot of research to do before I feel ready to start writing out the episodes, and so I'm going to give myself a pretty long break to be able to do that. For now, I'm setting a target date of October 3rd, 2026 to premiere Season 2. I'll aim to release some short episodes over summer to let you know what I'm working on, and I'll be experimenting with more video content, so make sure to follow me on YouTube at ModifiedPod. So before I say goodbye for now, here are your 5 take-home messages for this episode. Number 1. CRISPR-Cas systems are found in most bacteria and are an immune system to defend against viruses. Number 2. Cas9, together with a guide RNA, acts as a programmable DNA endonuclease, which can make DNA cuts at target sites based on a 20-nucleotide spacer region in the guide RNA. Number 3. CRISPR as a tool for gene editing was first demonstrated in 2012, and it immediately generated excitement. However, for a mix of reasons, commercial products are now only just arriving on the market. Number 4. A DNA break will in most cases result in random mutations at the break site, but if a DNA repair template is present, it will sometimes result in the template sequence being inserted at the break site. This is called gene editing. Number 5. Gene-edited crops face an uncertain regulatory environment currently, but in many countries, they are more lightly regulated than conventional transgenic crops. Well, Season 1 has been a blast, and it's time for me to get right to work on Season 2. Bye!